Hello everyone, welcome to my channel Biofingerprints. So in our last video, we had covered many topics like basic structure of a cell, discovery, contributions of biologists, etc. So today we will continue and start from the cell organelles. But before this, I request you to please watch my previous video. The link is in the description box. So today we are going to cover all the cell organelles, their structure and function. So our first cell organelle is endoplasmic reticulum. So it is a network of folded membranes. If you could see, here are some folded membranes. So it is the network of some folded membranes. Folded membranes. And these folded membranes are known as cisternae. These are not cristae. Cristae is a part of mitochondria. This is cisternae. Cisternae in endoplasmic reticulum and cristae in mitochondria. So the folded membranes are known as cisternae. Together they are known as cisternae. So it is the network of folded membranes. So there are two types, rough and smooth endoplasmic reticulum. So rough endoplasmic reticulum, when ribosomes are attached on the surface of endoplasmic reticulum, it is known as rough endoplasmic reticulum. So here in this image, could you see some beads like structures? So these bead like structures are ribosomes. And yes, the presence of ribosomes over the surface of ER makes it rough. Right, so therefore I am repeating once again that when ribosomes are attached on the surface of ER, endoplasmic reticulum, it is known as rough endoplasmic reticulum. Right, and what is smooth endoplasmic reticulum? It means when no ribosomes are attached. Could you see there is no such bead like structures are present, right? So no ribosomes are attached on the surface of ER. So then it is known as smooth endoplasmic reticulum. So at outer end, ER is connected with the cell membrane. So this is the cell membrane. So it means this is its outer end, because endoplasmic reticulum at its outer end, it is close to the cell membrane, right? And at the inner end, endoplasmic reticulum is connected with the nuclear membrane or the nuclear envelope. So this is its inner end and this is its outer end. How do we know? As at the outer end, ER is connected with the cell membrane, and at the inner end, ER is connected with the nuclear membrane, right? Now come to the functions of endoplasmic reticulum. So these are separate functions for SER, smooth endoplasmic reticulum, and rough endoplasmic reticulum. But here is a collective function of ER, that is, it forms supportive framework of the cell. Supportive framework of the cell. So it is also involved in the distribution of various substances among different parts of a cell, right? So the functions of smooth endoplasmic reticulum are enzyme and lipid synthesis. Lipid is the form of fat, production of hormones, and microtubules. Microtubules are the structures involved in cell division when cell divides. Next, it also detoxifies drugs and poisons in our body. Next, coming to the functions of RER. RER provides surface for ribosomes. As we have already discussed that if ribosomes are present on the surface of ER, it makes the surface of ER rough. Next, moving on to lysosomes. Lysosomes are also known as intracellular digestive centers. Why? Because here intra means within, and here cell is simple, the cell. It means within the cell, some digestive enzymes are present which destroy and digest foreign substances around them, right? And foreign substances could be like cellular waste or viruses or bacteria or any other pathogens or allergens, right? And yes, if a cell is fully damaged, it means it cannot be recovered or repaired. Then lysosomes help it to self-destruct. Okay, and this process of self-destruction is known as apoptosis, which is also known as programmed cell death. Programmed cell death. Right? So therefore, they are also known as suicidal bats. So now moving on to mitochondria. So it is a double membrane bond structure. So double membrane bond structure, it means it has outer membrane and inner membrane as well. And it is also known as powerhouse of the cell. Why? Because it provides energy. It provides energy to the cell, but in the form of ATP molecules. ATP, adenosine triphosphate. I am writing here the abbreviation adenosine triphosphate. And yes, this is also known as energy currency of the cell. This ATP molecules are also known as energy currencies of the cell. So there are two membranes in mitochondria, outer and inner. I have already told you. So the inner membrane, this membrane, okay. So this membrane has some infolding. So if you could see, there are some infoldings, right? So these infoldings, they are in finger-like projections. They are finger-like projections known as cristae. Please don't get confused with cisternae, which is the inner folded membrane of ER. We have already discussed, right? So cisternae in ER and its cristae in mitochondria. So what is cristae? I am repeating once again. The inner membrane has some infoldings, and these infoldings are the finger-like projections. Basically, they are known as cristae. Now come to the function of mitochondria. We have already discussed that it provides energy to the cell in the form of ATP. So therefore, this is known as powerhouse of the cell. And number two, it is also a site of aerobic respiration. Aerobic respiration. And yes, one more important thing, that mitochondria and chloroplast, these are the two cell organelles which have their own genetic material, right? They both are double membrane bond structures and both have their own genetic material, mitochondria and chloroplast. Next cell organelle is ribosome. So this is the structure of a ribosome. Ribosomes are found in both prokaryotes and eukaryotes. Next, ribosomes are attached over the surface of endoplasmic reticulum. That also we have already discussed. See, I always say that this we have already discussed, that we had already discussed. So therefore it is suggested not to skip video in between, right? So in ribosomes, RIB, these three letters. So RIB comes from RNA, its abbreviation ribonucleic acid. Ribonucleic acid. So here RIBO. So RIB, three letters are originated from here. RIB, ribonucleic acid. Next, so this RNA is the precursor for protein synthesis. Right? Next, ribosomes are not surrounded by any membrane. Next, if you could see in this diagram, there are two ribosomal subunits. One is large and another one is small. This is small and this is large subunit, right? So these two subunits, when they combine with messenger RNA, mRNA is messenger RNA, here M stands for messenger and here T represents transfer RNA, right? So when they combine with this mRNA, messenger RNA, they start protein synthesis. Now come to Golgi apparatus. It is also known as the delivery system of the cell. Why? We'll let you know. Delivery system of the cell, right? So Golgi apparatus, these are the tubular structures. If you could see, there are some structures present over here which are tube-like, right? So these are tubular structures, and and Golgi apparatus is formed by the ER, endoplasmic reticulum. Actually, what happens when, we can say that Golgi apparatus is originated from the ER. Actually, what happens when ER releases vesicles? Vesicles are some small structures which are released by ER and later they form into the membranes known as Golgi apparatus. Collectively, the structure is known as Golgi apparatus. So, Golgi apparatus is located near the nucleus. And what is the main job of Golgi apparatus? Golgi apparatus, it helps process and package proteins and lipid molecules into vesicles for delivery to target place or the destination. So therefore it is known as the delivery system of the cell. It means it delivers many substances like proteins, lipid molecules. Next, plastids. So plastids are present only in a plant cell, right? It is involved in photosynthesis. Plastids are of three types: chromoplast, chloroplast and leucoplast. So here chromo, their suffix indicates that chromo means color or colorful, and chloro means green color, leuco means white, so white in color, right? So chloroplast are the structures that have green pigment called as chlorophyll. So chloroplast, we'll discuss more about chloroplast in detail later, right? So chromoplast, first chromoplast. So next is chromoplast. So chromoplast, here chromo means color. So it helps in attracting insects for pollination, okay, because it provides different colors to the plant or flowers, right, like red or orange pigment. Okay. So leucoplasts, these are white colored plastids. So they are colorless and they are responsible for providing storage of starch to the cell, and they are present in the cell of potato. Next, chloroplast structure. So chloroplast is a main site of photosynthesis, and chloroplasts are also found in plants. Chloroplasts are found in plants and algae. Chloroplast is a double membrane bond structure and have their own genetic material. I have already told you when we were discussing mitochondria that there are two cell organelles which have their own genetic material and they both possess double membranes. So chloroplast is one of the cell organelles. So this is the internal structure of chloroplast. Here stroma, it is the fluid filled space in the chloroplast. If you could see this light green colored space, this is stroma. So it is the fluid filled space in the chloroplast, which has chloroplast DNA, ribosomes and starch and many other enzymes also. So stroma carries enzymes, starch, DNA of chloroplast and ribosomes. Next, thylakoids. So these are discs. If you could see, these are some discs or coin like structures in chloroplast. So they are arranged in a stack or piled over one another, which is known as grana. So many thylakoids together are known as grana or granum. So what is the main job of thylakoids? Since in the membrane of thylakoids chlorophyll pigment is present, so it is involved in photosynthesis. Next cell organelle is vacuole. So vacuole is present in both plants and animals, but here is the difference. Both plants and animals, and it is prompt in plants. It means it is larger in plants. It is larger in plants, so it pushes the nucleus to the side in them. If you could see in this diagram, this is the structure of a plant cell and this is the structure of an animal cell. In a plant cell, this larger cell organelle, this is vacuole in plant cell, and this is a vacuole in an animal cell. So you could see the difference between both the types of cells, plant cells and animal cells, that in case of plant cells vacuole is more prompt, right, or more bigger. Now coming to this layer tonoplast, it is covered by, so the vacuole is covered by an outer covering which is known as tonoplast. So tonoplast is the outer covering of the vacuole. And yes, this vacuole is filled with water or sap. So the content of the vacuole is known as cell sap, right? And the presence of water inside a vacuole makes it turgid or in swollen state. So we have already discussed this condition, turgidity condition, in our previous video, right? So please see it in our previous video, the link for which is in the description box. Next, come to the functions of vacuole. It functions as an excretory organ and also stores water. Now, how can you differentiate between a plant cell and an animal cell on the basis of the presence or absence of some of the cell organelles? So, in case of plant cell, centrioles are absent, but they are present in animal cells. Cell wall is present in plant cells only. It is not present in the animal cells. Chloroplasts are present only in the plant cells since they are involved in photosynthesis. One big vacuole or prompt vacuole is present in plant cell only, not in an animal cell. Cytoskeleton is not in your syllabus. Let's do an activity, right? So this is regarding the identification of various cell organelles in both the types of cells, plant cells and animal cells. So if you could see in this diagram, A is the plasma membrane, right? Since it is present in both. So in both, cell membrane or plasma membrane is present, but B is present in the plant cell only. So it means it is the cell wall because it is present only in a plant cell, and this is the cell membrane or a plasma membrane. Next, C, the dotted structure is cytoplasm, which is very common in both. And D is vacuole. How do we know that in case of plant cell it is more prompt or bigger in size? Next, E is nucleus. Since nucleolus is present inside the nucleus, right? And F is, since F is present only in plant cells, so it means F could be the chloroplast or plastids, right? And H is present in both, and this shape is somewhat like, say, internal infoldings. This is for mitochondria. So here H is mitochondria. Okay. Just for your information, mitochondria are numerous in case of an animal cell, but they are few in a plant cell. And yes, next structure is G. G is present only in an animal cell and not in the plant cell. It means it must be centriole. and centriole is involved in cell division. Now coming on the cell division part, cell division is the event, cell division is the stage when actually the cell divides. This is not in much detail in your syllabus, but yes, cell division is of two types, meiosis and mitosis. So in mitosis, when a parent cell divides, it results in two daughter cells, and in meiosis, when a parent cell divides, it results in two daughter cells. Then further two daughter cells divide into four daughter cells. So as a result of meiosis, four daughter cells are formed, and as a result of mitosis division, two daughter cells are formed. So now this was all about the fundamental unit of life chapter, where we have studied cell, shapes of cells, the purpose of some specific shapes in a cell, right, and cell organelles, the structure and function of various cell organelles. And in our next video we'll start new chapter, tissues. Till then, keep watching my channel. Thank you so much for watching. Please like, share and subscribe for upcoming videos. Thank you so much.